How crazy would it be to find out that you've had a bug living on you your entire life, and not just that, but thousands of them residing on your face at any given time? This is the mite Demodix follicularum that likes to reside in the hair follicles on your face. However, when the mite passes and the bacteria exits the mite, it is believed to activate your immune system and lead to neutrophil recruitment and activation of the pilosebaceous unit. This is your hair follicle, auricular pili muscle, and sebaceous gland all together. Why would this cause irritation though? It is believed that bacteria inside the mite can cause issues when the mites overpopulate and die off. These bacteria that reside within the mite are called Bacillus oreanus and help the mite to digest what they consume, such as sebum and dead skin cells. In 2013, researchers tested the theory that rosacea is caused by Bacillus oreanus by examining Western blot samples as well as the patient's reaction to the proteins from the bacteria. Western blotting is a technique to detect certain proteins in a sample of tissue or blood. In this example, it was tissue. Within this Western blot, protein samples collected from Bacillus oreanus were separated with SDS page, which separated the proteins according to their molecular weight. Proteins are negatively charged, which because of the matrix setup, the electricity that was conducted through the gel within the SDS page, proteins move from negative to the positive end of the gel. The smaller proteins will move faster than the larger proteins. These proteins, once separated, cause bands that correspond to the different size proteins. Proteins of similar size are able to occupy the same space of protein of interest. This leads to step 2 of Western blotting. The proteins are transferred to membranes. In this experiment, it was nitrocellulose membranes. The membranes allow for the specific protein of interest to be detected. The transfer was done by putting the SDS page gel and membrane next to each other in a transfer apparatus. These two items were electroblotted. Proteins from SDS page moved into the nitrocellulose membrane using an electrical current, making proteins move from the gel to the membrane. A buffer is used to avoid unspecific binding so that the protein of interest can be identified. A primary antibody that only binds to the B. oreanus proteins was added. A chemical and a secondary anti-human whole antibody sigma collected from patients was used. The appearance of bands indicated an immune response, or rosacea. This image is representative of the Western blots the researchers conducted. The bottom is indicating case number of the specific patient. The left is indicating the specific size of the proteins in kilodaltons. Lines at 83 and 62 kilodaltons is indicating an immune response, or rosacea. A, B, and C are displaying positive immune responses, with A responding at 83 and 62 kilodaltons, B responding only at 62 kilodaltons, and C responding at 83 kilodaltons. D and E have these lines absent, indicating a negative immune response. While the Western blot can be a useful device, it is qualitative as a standalone. To understand the population and test causal relationships, quantitative data is needed. This is where this graph comes in. It is a quantitative representation of the Western blots of the patients previously seen. The x-axis is showing the two groups within this experiment, the control not diagnosed with rosacea and those diagnosed with rosacea. The y is representative of the number of patients. The white bars are the total number of patients whose Western blots reacted at 62 or 83 kilodaltons or both locations. The black bars are showing the total number of patients within each group, this black bar being comparable to the control group and this one being comparative to the rosacea group. The control had 27% of its population react to the Western blot, while those diagnosed with rosacea had an 83% reactive at the Western blot locations. The stars indicate a significant value, indicating a causal relationship between rosacea or immune response and the bacterial proteins. This provides evidence that the researchers' hypothesis is correct.